Hey guys, today I'm going to be showing you how to make bread and then I'm going to explain some chemistry behind it. So the first step is to make the dough. This is kind of a fancy bread. It's called brioche. My mom's always wanted to try making it, so I thought it'd be a good opportunity to explain some of the chemistry. So here are the ingredients. The order in which you mix them and add them is important, but I'll be explaining that later in the analysis. The next step is to let the dough ferment in proof. So I did this by covering the dough and letting it rest overnight for 12 to 16 hours. Then after that was up, I kneaded the dough and then I cut it into 12 pieces. I arranged 6 pieces each in 2 loaf pans and I let them rest for another 4 to 5 hours before the last step where I baked the bread at 350 degrees Fahrenheit or 180 degrees Celsius. All right, let's get on to the procedure. The first step was to measure out all the ingredients, starting with the flour. The recipe gave all the ingredients in terms of cups and grams, but luckily I had a scale, so I could use some units we are more familiar with. So I measured about 142 grams of flour. Next, I measured out 4 grams of instant yeast and mixed it with the flour until I had a homogeneous dried mixture. I then tared out a ramekin and measured 40 grams of sugar and 44 grams of whole milk. I then whipped up 3 whole eggs and then I added all of those components to the dry mixture, then broke out the hand mixer again and I mixed the dough until it was stretchy and homogeneous. The next step was to chop up some butter and mix it into the dough. The recipe called for 115 grams, so I just used half of that pre-weighed stick. I then kneaded the dough by stretching it and pulling it. Then I placed it in a bowl and I covered it with saran wrap to rest overnight. As you can see, quite a bit of pressure had built up overnight, but we'll get to that later. After I had split the dough into 12 roughly equal pieces, I made an egg wash and then I brushed each of the pieces before letting them rest for another 4 to 5 hours. Once they had reached this size, I threw them in the oven for 20 minutes at 350 degrees Fahrenheit, which is about 180 degrees Celsius, and then I pulled them out once they were this nice golden brown color. I then sliced the bread up and made a really tasty sandwich out of it. Okay, now that we're done with the procedure, I think it's a good time to look at some of the chemistry. So traditionally, bread is made with a couple of key ingredients, so water, yeast, flour, salt, and sugar. But for this recipe, we didn't use any water, but we kind of substituted for it by using 3 whole eggs and then whole milk. So eggs are made of 74% water, and whole milk is made of 87% water. So the 2 proteins that make up gluten are gliadin and glutenin. So neither of them are really soluble in water, but when they are exposed to water, they have the chance to interact and form this protein matrix called gluten, which is very stretchy and elastic, in part because of the interchain interactions that occur between these 2 proteins. So one of them is these interchain disulfide bonds that are able to form in the oxidizing environment due to both of these proteins having cysteine residues in their primary structure, and we can see this covalent disulfide bond. And then there's also an extensive hydrogen bonding network due to the high concentration of glutamine, tyrosine, and serine residues in each of these proteins that, as you can see from their structure, have lots of moieties that are capable of hydrogen bonding. So I was talking about the importance of adding the butter last. And this is because butter has a lot of triglycerides that if it were added before we had mixed the dough and allowed this gluten matrix to form, these triglycerides would have the opportunity to coat these proteins. That's not a good place to put that. To coat these proteins and cause them to, and prevent them from efficiently forming this matrix. But when we add the fats last, they can kind of disrupt the gluten network without completely destroying it, which is kind of why fats are often referred to as shortening, because they shorten the gluten network and they make the bread softer and more tender. So one of the things I forgot was salt. I kind of tasted it, and it might explain why my bread wasn't that chewy. because salt is shown to slow fermentation and also increase the strength of the gluten network. All right, now I thought it would be a good time to take a look at the role of yeast in the leavening of bread. So yeast can acquire energy in the form of ATP in two ways, respiration and then fermentation. In both of these processes, carbon dioxide is released as a byproduct, which is kind of why we see the bread rise, because as this carbon dioxide is produced, it gets trapped inside of that gluten network that we talked about, causing the bread to expand. And it's also why I saw that increased pressure when I let it rest overnight, because some of that carbon dioxide is also just released into the air. So that carbon dioxide is just going to diffuse out of the bread as we cook it, because there's a high concentration of the dough, which is going to get released as we heat it. And then the ethanol that's also produced is going to evaporate under 180 degrees Celsius. So starch is one of the sugars that we find in flour, and it's a polysaccharide made of these glucose monomers that are linked by these alpha-1,4 and alpha-1,6 glycosidic linkages. And then we also added sugar, so sucrose. Yeast has enzymes that are capable of breaking both of those down into glucose monomers that it can actually use in this reaction to acquire ATP. So invertase takes sucrose and turns it into glucose and fructose, and then maltase is able to break down starch into glucose. So one of the things I noticed was that the brioche dough wasn't quite as airy as this experimental dough that I made with just the ingredients, just the key ingredients that we talked about earlier. So one of the reasons I kind of can hypothesize is that in the brioche dough, the yeast have access to a lot of different substrates, and a lot of them where they can't use. So we have these fatty acids that you can find in milk, and there's also casein proteins, and then we have proteins and fats that you can find in eggs that yeast do not have the enzymes to break down and convert into usable energy. Whereas in the classic bread, the yeast are really only surrounded by sucrose and then starch, which, as we talked about, it can convert into glucose so that it can produce carbon dioxide and acquire ATP, which is why we see the lighter, gassier bread when you only have flour, salt, water, sugar, and yeast in the dough. This is also kind of why it's important to mix the dry and wet ingredients separately, because when you mix the dry ingredients, you can kind of ensure that the yeast and the flour are evenly distributed so that the yeast aren't just concentrated in one area. The last thing I wanted to take a look at was the Maillard reaction, which is responsible for the browning of the crust that we see as we bake the bread. So here's a generalized mechanism where we have an amino acid acting as the nucleophile and the linearized sugar acting as the electrophile. So we see the formation of a tetrahedral intermediate. Then we see this shift space, which upon treatment with base becomes this enamine/enol, which undergoes tautomerization, and then we get this ketone, which is susceptible to intramolecular attack by this hydroxy moiety, and then we get this glycosamine. So the glycosamine then undergoes an amidori rearrangement and forms a bunch of ketosamines, which are then able to degrade into a bunch of different products like reductones, fission products, and then these nitrogen polymers that show up as this nice brown color. So one example is melanidins in bread, which is why we get that nice crust. Here are a couple of other products of the Maillard reaction. We see different aromas and tastes that come out of this, very general reaction that can lead to lots and lots of different chemicals. And then here's one of the main proteins in egg, ovalbumin, that, as you remember, we did an egg wash over the bread to help facilitate this Maillard reaction so we can get an even nicer brown crust.